All right. I guess we have some people coming in. So as those people come in, I would like to welcome everyone to Discovery Day. Yay! I do, before we start, want to give some special thanks to Tracy Kazmersky, who was sort of my partner in crime in trying to organize this, but good luck or bad luck happens to be on vacation with her kids today. So that's great. And then I would actually like everyone to give a round of applause to Becky Choff, who has organized amazingly. I spent the entire year organizing everything. So please give her a round of applause. Compliments all go to her, complaints come to me. And then just to remind you, there is a survey outside. There's a QR code for a survey, so we'd like to get some feedback. So welcome everyone to the 5th annual Discovery Day. Discovery Day evolved as it was originally the K-12 retreat for those of you who are under the K-12 or eventually want to get support from the K-12, and that is ultimately under the Children's Health Research Career and Development Award. The broad vision is to really sort of showcase junior faculty who want to be physician scientists. So how many of you out there want to be physician scientists? How many of you out there are actually curious about your patients, about their disease process, how it works, what the mechanisms are, what the potential treatments are? How could we make treatment better? So we're all scientists in some way, shape, or form, right? We all are curious. That's why we went into medicine. And you don't have to feel like you have to be a physician scientist. All you need to be is curious. So this Discovery Day really allows junior faculty, residents, fellows, and other trainees to really engage in diverse areas of discovery. But physician scientists aren't just about doing bench work. It's also about being a physician, asking clinical questions, showing the data. We want to make sure that the things aren't pigeonholed. And so this retreat is really to have an invited speaker who is an outstanding physician scientist and mentor, who I'll introduce, but also for you to hear about the types of science from our trainees. I would just ask, I know a lot of times we see grand rounds titles and we say, oh, that's not for me. This is going to be too hard. I'm only going to understand half of it. I want you to open your mind. I want you to be curious and I want you to listen about what Dr. Di Paola is going to say. So Dr. Di Paola I just met him last night. He's a great person. And it just so happens that Alejandro might be his sort of neighborly evil twin. They both were raised in Buenos Aires and have a lot of similarities. So he completed his medical school in Buenos Aires and then came to the United States to do both residency and a HUMOC fellowship at Orange County, California Hospital, Children's Hospital, and then moved to Iowa. And it was while in Iowa, he was actually under the CHRC-DA mechanism to actually begin to learn about science and sort of hone his skills. While in Iowa, he became an associate professor with tenure and was in their interdisciplinary program in genetics. He then became faculty in the Human Medical Genetics and Genomic Program at the University of Colorado. He then, in 2019, moved to WashU, where he became the division chief in HUMOC. And is currently the Elizabeth Finney McDonald Endowed Chair of Pediatric Hematology Oncology, and he's also the Associate Director of their MSTP program. He is a former president of the Hemostasis and Thrombosis Research Group, as well as the former president of the American Society of Pediatric Hematology Oncology. I could go on about the number of successful trainees he's had and the years and decades of independent funding that he's had, but that would sort of detract from having him from Discovery Day. So if you could all please welcome Dr. Di Paola.
Good morning, everyone. Thank you, Lane, for that kind introduction. I wanted to thank Terry and Alejandro for inviting me, and Lane for inviting me to this day. I'm really excited and honored to present our research in your Pediatric Discovery Day. As Lynn was saying, I was a CHRC scholar. That was 20-something years ago. I used to have a lot of hair, and I was 145 pounds at that point. Things changed a little bit. But over the last years, then, we have, as Lynn was saying, we have set up a research program in my laboratory, and then that have been traveling with me, whatever places I go. And it's really nice to see familiar faces in the crowd here. Okay, so as Lynn was saying, I know that there are a lot of, there are many people that are not Pitt's hemonc, so I'm gonna try to adapt to that, and I hope that you can enjoy our approach to these hereditary bleeding disorders and from the laboratory point of view. And so today, what I'm gonna talk is, I'm gonna give you a brief overview of normal hemostasis, platelet and factor production and function. I'm gonna take you back to your medical school years and remind you why this is important. Then I'm gonna talk a little bit about inherited platelet disorders of von Willebrand's disease and its clinical presentation. And then I'm gonna give you 2 clinical cases where we as a lab kind of try to decipher these clinical and genetics conundrums. So this is a mesenteric blood vessel in a mouse, actually. It's not a human, it's a mouse. And as you can see there is a pronounced or a repetitive injury. You can see the blood coming after in the extravascular space. And then, but you can see a clot forming here. And that clot, if you keep looking, is gonna be actually remodeled over time. And it's gonna kind of disappear, at one point, leaving a wound in the blood vessel. Of course, this is a model of hemostasis, but that's what we think that occurs in our blood vessels daily, and many of our blood vessels, where the constant repair of blood vessels by the hemostatic system, is happening. So as a lab, we decided to study, this is a very complex phenomenon, but we decided to study 2 axes of this phenomenon. One is platelets, and the other one is von Willebrand's factor. So what happens there in that, that image that I just showed you? Well, as I said before, if you have an injured blood vessel, as you can see there, you have von Willebrand factor that is exposed in the subendothelium. These are endothelial cells. You have von Willebrand factor, of course, because it's our preferred molecule, it's giant here. And then you have platelets, okay? So what happens, when you have this collagen exposure, then you're gonna have the formation of a platelet plug, and that's gonna trigger the coagulation cascade. And the coagulation cascade main objective is to cleave prothrombin into thrombin, which is the most potent procoagulant protein in your body, and then eventually cleaves fibrinogen. And it does that through a series of proteases reactions where by gene duplication, evolutionary biology has adapted into these really complex protease cleavage of each one of these factors, as you can see there, that eventually, as I said before, is gonna cleave prothrombin into thrombin, cleave fibrinogen and make a fibrin clot. And eventually, like any other system, you have anticoagulant proteins that are counteracting into this prothrombotic phenomenon, including protein C, protein S, and antithrombin. And then, of course, you have a fibrinolytic system that is gonna remodel that clot, as you saw there in that animal model that I showed you, and it's gonna remodel that clot into this wound that is gonna fix the endothelium. So as you can imagine, this balance between bleeding and clotting is pretty fine. And there are many gene and gene variants that are involved in that. And actually, over the last 30 years, first by positional cloning and then by next generation sequencing, most of the genes that are related to these components of the system have been discovered. So our laboratory that started, as Lynn was saying, 20-something years ago in Iowa, by, I trained in human genetics, by a combination of human genetics and animal models, we tried to study disorders of platelets and von Willebrand factor. And we did that by studying families that have platelet dysfunction or von Willebrand's disease, with the idea that if we discover genes that are associated with these diseases, then we can apply the same genetic knowledge to quantitative traits that are included in von Willebrand factor levels or platelet function or platelet volume, for example, that are involved in cardiovascular disease. And I remind you that cardiovascular disease is still the main killer in the world, the #1 killer in the world. So that was our idea when we started our laboratory many years ago. So before I go over, so where are platelets and von Willebrand factor made? I just want to remind you, for those that do not do Pitt's hemonc or hematology, that this is a very simplified version of hematopoiesis, where you have hematopoietic stem cells in your bone marrow. You generate these progenitors that are differentiated from these cells. This is a megakaryocyte erythroid progenitor, and that progenitor, we believe, again, in this very simplified version, that is gonna give rise to this very fascinating cell that is the megakaryocyte, and it creates these proplatelets that are gonna go into the sinusoids and make platelets, and then into erythroblast that is gonna become reticulocytes and eventually red cells in the periphery. And in order to do that, you need a constellation a constellation of transcription factors that are gonna be differentiated these cells in many different pathways, as you can see there. And this is relevant to one of the cases I'm gonna show you. And then you do that in order to maintain or to generate 10 to the 11 platelets a day, actually, and to maintain a platelet count that is approximately between 140 and 450,000 per microliter. Okay, so that's how platelets are made. So how is von Willebrand factor made? So the simple thing about von Willebrand factor that is only made in two cells. One is the megakaryocyte, as I shown you in the bone marrow, and the other one is the endothelial cell. So as you can see here, you have in this schematic, you have endothelial cells that have these granules that are called wavy polarity bodies, and they secrete von Willebrand factor into the circulation by two different mechanisms. And they get, as soon as it comes out of the circulation, it's cleaved by this metalloprotease called ADAMTS13. So von Willebrand factor is gonna either travel in this globular form, carrying factor VIII, coagulation factor VIII, to the point that is so important for coagulation factor VIII survival that if you don't have von Willebrand factor at all, you have hemophilia because factor VIII gets cleared from the circulation. And then it's also traveling in platelets. As I told you before, megakaryocytes make von Willebrand factor and it's traveling in the alpha granules of platelets. And then it gets cleared by some mechanisms that we still do not understand well. So how do they work? We know what they're made, how do they work? Well, as I said before, if we over the last 30 to 40 years, scientists and our lab included have figured out how this works in terms of the molecular signatures that are important for this adhesion to the subendothelium. So you have subendothelium. Again, these are all simplified versions of schematics with von Willebrand factor, collagen, laminin, and extracellular matrix. And then you have platelets that by its receptor of von Willebrand factor are gonna tether, when they see that exposure. Then you have other integrins and other receptors that are that facilitate adhesion. And that can be seen in this really nice scanning microscopy that Katrina Bark took in my lab. As you can see there, platelets become almost like a fried egg on top of the collagen. And then you're gonna have platelets accumulated on top of each other via the integrin alpha-2b beta-3 or fibrinogen receptor. And you're gonna have that process that I showed you before, which is the formation of the fibrin clot, okay? So what happens at the platelet molecular level? And again, this is important because this is the way that we understand how to test for these disorders. So you have platelets, you have a platelet there with several receptors. And then if you have an stimuli, so you're gonna have that either collagen or thrombin is gonna activate the platelet. As you can see there's calcium mobilization. And what happens is something that is called inside-out signaling. As you can see there, this integrin changes conformation, it's gonna bind fibrinogen. And fibrinogen is not only allowing to bridge the platelet to each other, but also signals or triggers a signaling cascade called outside-in signaling. And you can detect this by the platelet aggregation that some of you order, from the lab. So when you have that outside-in signaling, you have a spreading changes, as I'm showing you before. You can detect that by the scanning microscopy. You have granule release, as you can detect by flow cytometry. And then you have the ultimate event, which is the flipping of the membrane and getting that docking site for the clotting factors to generate thrombin and what we call procoagulant activity, and we can measure that also by flow cytometry, okay? So how is this translated into clinic? Actually, as I showed you, this is a complex, very complex phenomenon, but clinically it's very, I wouldn't say simple, but it's very distinctive if you have deficiencies in clotting factor, if you have deficiencies in platelets of von Willebrand factor. So if you have deficiencies in the coagulation cascade, most of the phenotypes are gonna be deep bleeding into hemarthrosis into joints. And then you're gonna have deep muscle hematomas, so it's the pattern of hemophilia bleeding or other coagulation deficiencies is very similar usually. But if you have deficiency of platelets of von Willebrand factor that give that primary hemostasis that we were talking about, then you're gonna have mostly bleeding represented by uterine bleeding, by GI bleeding or by skin bleeding or bruising. Okay, so the phenotype patterns are very different. And therefore, the clinical presentation of platelet and bone factor deficiencies is mostly mucocutaneous bleeding, represented by nosebleeds, excessive menstrual bleeding, easy bruising, post-tooth extraction bleeding, and post-surgical bleeding. So when do we suspect as clinician that we have an inherited platelet disorders? Because I'm sure that hematologists here in the audience, or even people that do ICU, you consult hematologists a lot for thrombocytopenia. So when do we think, because we're gonna talk about congenital thrombocytopenias, why do we think that we have a congenital thrombocytopenia or hereditary? Well, the persistence of neonatal thrombocytopenia, of course, you know, if your patient that you saw in the NICU keeps having low platelets during the infancy, then you think that probably there is a hereditary bleeding disorder. A family history, of course, of thrombocytopenia, these are fairly intuitive. Mucocutaneous bleeding or bruising that is usually out of proportion of the platelet count because a lot of the thrombocytopenias are associated with platelet dysfunction. The presence of associated clinical laboratory features of the hematologists here, or if they are hematology fellows or hemonc fellows, they know that we always emphasize about skeletal dysplasias or some other things that could be associated with platelet dysfunction or platelet issues. And then the most common form probably is that when we treat ITP or immune thrombocytopenia purpura that is refractory to all treatments, and eventually we send a genetic panel and voila, you know, we have a mutation. And then of course, as we say, always failure of typical causes of acquired platelet-related bleeding that account for the systems. So as I said before, over the last, I would say 25 years, 30 years, we have kind of set up the map for the genetic associations with platelet disorders. And we can, that's a megakaryocyte, and we can group them in of course, in different families of mutations that make sense physiologically. But more importantly, I would say for the clinician, is that all these disorders, as you can see here by this complex Venn diagram, and you have the genes in the middle, are sometimes associated with syndromes. And you have to think about these things because these gene deficits or these gene mutations can not only give rise to platelet disorders, but also to other syndromic associations with these particular disorders. So how do you work them up? You know, and again, this, you don't have to remember this, but just to give you an idea that if you have a patient like this with mucocutaneous bleeding, we have some sort of algorithms that we can use in order to initiate the differential diagnosis. And usually what we do, you know, of course, the hematologist in the audience can tell me if they do differently. We do this initial test. If we have a normal platelet count, we do usually, or maybe at the same time, von Willebrand factor levels. If they're normally low, we call that von Willebrand's disease. If they're normal and we think that the patient has a phenotype that is compatible with the platelet disorder, we do platelet aggregations. And of course, if the platelet have low platelet count, we follow that path of diagnosis. And it's this more complex algorithm. You can follow the path of thrombocytopenia in your differential diagnosis. And of course, you know, the ultimate functional test that we use in the clinic for platelet dysfunction is platelet aggregation, which I remind you that it also shows the failure that we have implied over the last 50 years in getting up with a better test. You know, platelet aggregation is 50 years old, and we haven't come up with a better test to determine platelet function. So as I said before, after this introduction, I'm gonna tell you a couple of cases where our laboratory, you know, tried to really dwell into the genetic causes of these diseases. So case one is a 3-year-old girl with a history of congenital and familial thrombocytopenia. That was interesting to us. We collect a lot of families, we recruit a lot of families. This family was from Detroit, and a friend of mine, Mike Callahan, called me and said, you know, this family is more interesting because there are people in the family that develop leukemia, not only just thrombocytopenia, but leukemia. As I said there, interestingly, her maternal aunt has some thrombocytopenia and previous LALL. Several members of the family have thrombocytopenia. And at that point, they sent a gene panel that had most of the genes that are at that point associated with thrombocytopenia and leukemia, including RANX1, for example, and that was negative. So there were no cytogenetic abnormalities in the leukemia samples. So I convinced Leila Notsley, who was a PhD student in my lab, to have a side project, actually, that you'll see, interestingly, it turned out to be her main project, of course. And because we really got interested in these families, you can see there, the interesting thing about these families, not only this is an autosomal dominant disorder, at least what it looks by the pedigree here, but there are two cousins that have the phenotype, which in this case is low platelets, okay? And when two cousins share the phenotype, you know that by definition, they're gonna share approximately 10% of their genome. So eventually we say, well, we can always, focus on that 10% and see if we can find the gene. So their phenotype was a platelet count that was, it seems like it was low in all those affected. And also they have a mean corpuscular volume elevation. Their red cells are bigger, okay? So when we went to their bone marrow, actually, we saw signs of what we call, you don't have to remember this, but we saw signs of what we call myelodysplasia. These cells are abnormally looking. And actually, if you see here on this one, we have an increased number of megakaryocytes, the cells that are making platelets. But when you look into those megakaryocytes, they are very hypomorph and they're abnormal, okay? So we did whole exome sequencing in 11 members. And you had to be lucky doing these endeavors. We sequenced thousands of people. And believe it or not, after sequencing 11 members of this pedigree, and we put this in the pipeline, the bioinformatic pipeline, we only came with one deleterious mutation in a gene, in the coding region of a gene, that's segregated with all the people that had the phenotype. And that was a heterozygous mutation on the gene called ETV6, which is a transcription factor. And it was a missense mutation, as you can see here in the depiction of the gene. This is the point, this is the protein, the pointing domain. This is the DNA binding domain as a transcription factor. And the mutation was in the middle of the central domain, which is a kind of a floppy protein domain that has not been crystallized. So what is ETV6? Well, as I said before, it's a transcription factor. We know that it dimerizes and maybe polymerizes, and there is no consensus nuclear localization signal on this. And this is important for what I'm gonna show you in the phenotype. So we published this, and I put this slide because it's interesting, because every time that you think, for the young people in the audience, that you're doing something that you are so unique, of course, there's somebody doing it too. And at the same time that we send the paper to Nature Genetics, we get a call from the editor that they're gonna publish this other paper from actually a good friend of mine, Akiko Shimamura. So these papers came at the kind of the same time. And then there was another paper that solidified this observation that ETV6 germline mutations are associated with thrombocytopenia and predisposition to malignancies. And then more interestingly, the St. Jude group that had germline on their patients as well as somatic tissue, they took 4,000 patients that developed leukemia, unrelated cases, and they did their germline for ETV6. They tested, and approximately 1% of these patients have mutations in ETV6, solidifying the fact that this is probably a cancer predisposition syndrome, and that the WHO, this is what my people in my lab claim is our claim to fame, change the classification based on this new cancer predisposition syndrome. So over the years, we have collected many patients. And what I can tell you about ETV6 mutations is like the penetrance for and this is important because we'll talk about later about the genetic counseling, but the penetrance for platelet count is almost 100%. So if you inherit the mutation, your platelet count is gonna be low. And for the MCV, the same thing. But the penetrance, I remind you, for leukemia is only 25%. I mean, it's not only, it's 25%. So 1 out of 4 are gonna develop leukemia at one point. And as you can see there, the mutations are very much distributed all over the gene, mostly in the DNA binding domain. So we know, because we did overexpression systems in our cell system where we do CD34 cells, we overexpress in a lentiviral vector. And then, as you can see here, if you have here, this is the cell, you have the DNA here. An antibody against ETV6 shows you that ETV6 is mostly, as most transcription factors, located in the nucleus. And as you can see here, the same. And if you tag ETV6, also is located in the nucleus. But when you put any of these mutations into the system, it seems like ETV6 comes out of the nucleus, okay? And it has this extracellular localization. Maybe that's the mechanism why it's not working well. So we came with this working hypothesis where if you have ETV6, you have normal hematopoiesis and you make good platelets. if you don't have ETV6 in the nucleus, you have bone marrow dysplasia, and you don't make any platelets, or you don't make good platelets, and you have increased your risk for leukemia. So this happens in an overexpression system. So a student in my lab at that point, Marley Fisher, who is an MD PhD, now is a resident, and you would think that she would be a resident in pediatric hematology, but she's a resident in dermatology. This is what we do to our students in our lab. Decided to see if that happened in vivo in these patients. So these are samples from patients, and as you can see here, ETV6 is mostly, we have a good antibody for ETV6. The mutated ETV6 is mostly in the peripheral blood cells, and this patient is outside in the cytoplasm. So we knew that ETV6 made the complex, by previous research, made the complex with HDAC3, histone deacetylase 3, and NCOR2, which is another complex, which is kind of a repression complex that goes and binds chromatin. And eventually she tried antibodies against these two members of the complex, and we had the same thing for both, for HDAC3 and NCOR2. So if this is a transcription factor, we know the transcription factor gets out on the cytoplasm, and that has been shown by other groups too. What are the transcriptional consequences? So we did, because we can, and it's out there, we did single cell RNA-seq of all these cells in the periphery of these patients. And as you can see here, the only thing that you have to see here is that this complex Venn diagram for all the nucleated cells, these 22 genes that are involved here in all the cells are differential expressed, that are upregulated in patients that have this mutation. And as you can see here in this, list, most of the genes are pro-inflammatory interferon response genes, actually, indicating that there is a dual repression of these genes, and ETV6 was calming this repression, or was repressing interferon response genes. But interestingly, if you put all these unbiased data into the pipeline, and you say to these GO genes, the GO term genes, you say, okay, what do they take me? Well, if you do ingenuity pathway analysis of all these transcripts, they all end up in HDAC3, indicating that probably the way that ETV6 is working, or is not working, is because it's not binding, or is taking HDAC3 out of the complex. So we thought, okay, so they have the repression of interferon response genes, these patients might be inflamed. Well, when we did cytokine expression in the periphery, there is no cytokine differentiation between the controls and the mutated one, indicating that probably this is a bone marrow phenomenon, and I'll show you in a second why, and not necessarily a peripheral blood phenomenon. So then what she did is she used a knockdown technology to knock down ETV6 in these peripheral mononuclear cells, and she obtained the same phenotype. So if you just take peripheral mononuclear cells from a normal donor and you knock out or knock down ETV6, you're going to have the de-repression of this interferon response genes and pro-inflammatory phenotype. So we came now to our working hypothesis on this is that if you have ETV6 with HDAC3 and NCOR2, you have normal hematopoiesis, you have normal megacardiopoiesis and thrombopoiesis, but if you have the ETV6 either mutated and it doesn't bind to DNA, or is outside of the nucleus and then it does not bind either, you have interferon response gene repression, bone marrow dysplasia, and then you develop a 1-point leukemia, okay? So we do what we always do, and we made a mouse model with our collaborators by CRISPR-Cas9. We deleted the murine ETV6, and we put a new gene that is murine actually, and this is important, that has the mutation in a different position, but it's the same mutation. And we were very excited when we saw that they have hyperplasia of megakaryocytes. They have more megakaryocytes in the marrow. But then I put here in a summary of the bone marrow of the animal models for these. The animal models haven't been as robust, or they haven't replicated well the disease, unfortunately. But as you can see, that there are two out there, one by Kim Nichols in St. Jude and one by us, that we just published in Experimental Hematology. That the only thing that I can tell you about this is that all of them seem to have this de-repression of interferon response genes, abnormal cytokine release in the marrow, and therefore increasing inflammatory milieu in the marrow, and probably causing myelodysplasia and eventual leukemia. But that is still waiting to see if that is real. So If we go back to the case, so this is what we try to do with this case, try to really understand the physiology and the molecular biology behind it. But if you go back to the case, because we are clinicians, as Lynn was saying, we're clinician scientists. So patient received the diagnosis of ETV6 germline mutation, which is responsible for thrombocytopenia and predisposition to leukemia. Several members of the family received the diagnosis. Thrombocytopenia is almost, as I said before, 100% penetrant, but leukemia, unfortunately, is 25%. 1 out of 4 of these individuals are going to develop leukemia. And we don't have a good way, surveillance way, to really determine who's going to do this. So we can talk later in the discussion, but when you do this gene analysis, you're going to have to tell the family, yeah, we understand the cause of your thrombocytopenia, but by the way, you have a 25% chance of developing leukemia at one point in your life. We know a lot more about these leukemias, and they usually do better than other leukemias in treatment, but that's all we can tell them. And unfortunately, I would love to hear, I'm a bona fide oncologist because I'm board certified, but I haven't seen an oncology patient in like 20 years. Some people in the audience know that, so I would love to hear if they have a better solution for this. So in the second part of the talk, I'm gonna talk to you about the second case. And as you saw, that was kind of somewhat easy to us. It was a Mendelian disorder. As I said before, we were very fortunate that we found the cause of thrombocytopenia, and that some people found it at the same time. So what I'm gonna tell you now is a case 2, which is a much more complex genetic phenomenon. And I'm gonna tell you what we're trying to do to solve it, but we haven't solved it. So it's not like the other case. So this is a 14-year-old female that presents to clinic. This is a patient of mine with excessive menstrual bleeding. Her mother had similar issues and eventually had a hysterectomy. Several members of the family report increased bruising and bleeding. The labs, we do the same labs that I told you that we do when we have mucocutaneous bleeding. She had a normal platelet count and a normal CBC. Her von Willebrand factor levels were, however, low. Her antigen is 39 international units per deciliter. You can see the normal range there. And the rhistocytin cofactor activity, which is a functional assay of the von Willebrand factor molecule, was also low at 43. The von Willebrand factor sequencing, we did sequencing by one of the clinical genetic panels that are out there, does not show a pathogenic variant in BWF. And she's ABO blood group O positive. And you're gonna understand why this is important. So what is von Willebrand disease? I love this slide because von Willebrand disease was described in Finnish, actually in Finland, by a Swedish German doctor named Erik von Willebrand. And that was a 1926 paper. I have a copy actually of that paper. And it was finally translated to English 25 years ago or something like that. But I love this because he was an internal medicine doctor, by the way. He was not a pediatrician. But the index case was, this one was a 5-year-old girl that had excessive epistaxis, had epistaxis. And as you can see there, he hand drew it's a hand drawing of the pedigree. And it was in the family in the Alan Islands, close to Sweden. And as you can see here, which is, I put this pedigree because not only it seems like it's a autosomal dominant disorder, as you can see there, it does not seem to skip generations, but also the term, even though people might say, well, you have von Willebrand disease, that's a mild disorder. As you can see here, the crosses are several, and they're mostly females, because females in this, before we understood this diagnosis and before we had therapy for this, will bleed to death of excessive menstrual bleeding or postpartum hemorrhage. So BWF is a gene that we've been studying for the last 25 years. It's in the short arm of chromosome 12, and it makes it's a very large gene. It has like 52 exons, and it makes this very complicated, very complex protein. I put that image there because it has been redefined, all these binding sites for von Willebrand factor, that it wasn't enough to be complex in this monomer that is actually dimerizes and multimerizes. So it's one of the largest proteins that you're gonna see in your body, actually. So what do we know about BWF mutational landscape? Well, we know that if you have a quantitative deficiency, you have type 1. If you have a qualitative deficiency, we call it type 2. And if you have an absolute deficiency, we call it type 3. Okay, so if you see this molecule here and you see the types here on the y-axis, you can see that the mutations are fairly distributed all over the gene, but with some, particular for the type 2s, which are the functional ones, those are mostly reserved for some specific hotspots within the gene. So what are the genetic mechanisms of what we know of von Willebrand's disease? So there is kind of a Mendelian form. If you have a mutation in von Willebrand factor gene, you get the phenotype. But I'm gonna show you that 75% of the people that have this disease, we cannot find mutations in that gene, okay? So type 3, as I told you, is autosomal recessive. You have to be homozygous for deleterious mutation or compound heterozygous. The majority of mutation cause null alleles, not surprisingly. Usually we sequence the gene, but also we complement with ACGH. Now we do whole genome sequencing and we can detect the structural variants by that. And carriers, interestingly, it's a very interesting disease. Carriers of this disease, you would think that because they have less than 50% will have the disease, do not have the disease sometimes, so to make it more complicated. Type 2, as I said before, is usually caused by missense mutations, and you have the full protein some of the times, and then it doesn't function properly. And most of these mutations act via a dominant negative effect, okay? And then type 1, which is the most common one, actually the patient that I described, we will have put money, if we could bet on this, that she had type 1 von Willebrand disease, and actually she was diagnosed with type 1 von Willebrand disease. It's characterized by incomplete penetrance and variable expressivity. It means that even if you inherit the mutation, you're not gonna have the phenotype all the time. And even within families, if you have patients that have the same mutation, they bleed very variably, okay? So as I said before, only 40%, this is the percentage of type 1 subjects. I remind you, 75% of the patients that have von Willebrand factor sequence variation. As you can see by the studies, this is variable. And these studies are over-represented probably because they call variants any variants, not necessarily the pathogenic variants, okay? And I think that this can be explained by this. I want you to bear with me. You have here this, Evan Sadler put this very nicely. This is the relative risk for bleeding, okay, or thrombosis. And this is the von Willebrand factor levels in the population. I remind you that von Willebrand factor levels in the population are not normally distributed because of the blood type O. So by definition, if you have your von Willebrand factor levels are between 50 and 200, by definition, the mean should be 100, by statistical definition. But if you have blood type O, and you take all the blood type O patients, 45% of the population, their mean will be 75. So blood type O is a modifier, it's a genetic modifier of von Willebrand factor levels. Okay, that's the reason why this curve is not normally distributed. So as you come down in your von Willebrand factor levels, you increase the risk of having a mutation, of course, and then you get diagnosed with von Willebrand's disease, okay? And we know now that if you increase the levels of VWF with aging, your risk of thrombosis goes up, and it might be related to those von Willebrand factor levels. But again, there is a population here between approximately 30 and 50 that we call low von Willebrand factor. That 50% of them might not have a mutation in the von Willebrand factor gene, and they present to your clinic bleeding, some of them. So that's what we're trying to figure it out. I remind you, by definition, 14% of patients with blood type O will have a von Willebrand factor level that is less than 50. That by definition, by laboratory definition, is abnormal. And of course, there is not an epidemic of bleeding in 14% of the population. So having this as background, what we are studying now is trying to understand the genetic regulation of von Willebrand factor levels so we can answer the question what are the genetic causes of this disease? So in order to find genes or genetic variants for any genetic study, you need to have a heritable trait. So the heritability of von Willebrand factor levels is interesting. Family studies have put the heritability around 30%, some of them 80%, as twin studies, but it's hard to correct for environment at that point. And I would say that heritability for the trait is probably around 50%. And 25% of the variance, as I told you before, is explained by the effect of the ABO blood gene. And of course, environmental stressors may account for approximately 35%, because if I go for a run this morning, my von Willebrand factor levels might increase by 20%, for example. Or if you get a cup of coffee, or if you poke a child trying to do a STIME trial, their von Willebrand factor levels will be higher, okay? So what are the factors that influence the variability? And I have to dwell into this because these are very important for our studies. One is ethnicity. We know that individuals from population of African ancestry have and this is worldwide, it's not just African Americans, have significantly higher von Willebrand factor levels, up to 25% increase. And this difference we know now is not explained by the ABO genotype frequencies. And then the other one is aging. Increase of von Willebrand factor with age is about 1 to 1.5 international units per deciliter. So without effect on the bleeding phenotype sometimes. So we know that those two factors are important. So people, of course, have done GWAS studies, genome-wide association studies, where you study, at this point, more than probably 200,000 people, not in this representation, but overall, worldwide, for von Willebrand factor levels in the normal population. And this is what it does, it's to give you signals of genes, usually common variants, that are associated with the levels in the population. As you can see here, we were surprised by all the number of genes that have been associated with von Willebrand factor levels. But of course, ABO is the most important one, as I told you before. We knew that from like 40 years ago. And von Willebrand factor itself causes, is influencing the trait, okay? So the questions that we're asking now is, what are the genetic mechanisms responsible for von Willebrand factor levels in low von Willebrand factor individuals? Because eventually we want to understand, so hopefully we can do in the future, polygenic risk scores, for example, and determine if you inherit these variants and you bleed, you're going to have your chances of bleeding postpartum or post-surgery will be higher, for example. That will be the end point. And we're not even there, so don't get excited about it. So and then the question is, what are the genetic regulators of VWF in individuals of African ancestry? And we're doing something on that. And then are there structural variation or complex variants? And I'm going to tell you a little bit more on that because it's interesting to us. So in order to do this, we kind of piggyback on one point, and then we took the lead of this Zimmerman program for the biology of von Willebrand disease. This is a PPG, a program project grant from NHLBI that Bob Montgomery, the great Bob Montgomery, started the university 20 years ago, and we've been part of that. And eventually, upon his retirement recently, he was kind enough to give us the lead, and we just submitted the grant. We got a great score. If you are in council, please, at NIH, be merciful, because we don't want to, so it's a 700-page grant, just to give you an idea. But the beauty of this grant is that over the last 20 years, Bob has collected and recruited these, as you can see there, retrospective cohort of patients with von Willebrand disease, approximately 3,500 individuals. And then through another R1 mechanism that we have, we collected the prospective cohort with another 2,000 individuals. Because as I shown you, those GWAS studies are in people that have von Willebrand factor levels, but are healthy individuals. So we wanted to have people that have the disease to understand the disease better. And then you have follow-up studies, we have bleeding scores. So eventually we have a really nice cohort that we can study, and that's what we try to convince NIH to give us money for. And the other interesting part is that we have a very deeply phenotyped cohort that is represented there by the VELOS sample biospecimen management system, which is like a management system that they have at Versity. And at WashU at the Genome Center, we have an interface where we can match de-identified data with their genomes, or de-identified patients with our genomes. And not only we have that, but we have approximately almost 100,000 samples from plasma from these patients and DNA. And again, so we have all these exciting resources that we're studying. So I'm gonna give you just to finish, a couple of nuggets of what we're doing with these. One is, Brooke Sadler, actually Evan Sadler's daughter, who works in my lab, had this idea. I said, well, we have already sequenced the von Willebrand factor gene, like 1,000 patients with von Willebrand disease. If we put this in an unbiased, pipeline, we don't know what mutations they have whatsoever. We know that in other diseases, in other traits, if you have rare variants, the higher the number of rare variants, the more deleterious that trait is gonna be. So she asked the question, what about if we count unbiased, the number of rare variants defined by less than 0.01 in the general population in von Willebrand's factor? And actually she found that if you if you increase if you increase the number of rare variants, as you can see here, are the lower your von Willebrand factor is predictor of your von Willebrand factor levels. And you can see that even with pathogenic variants versus non-pathogenic variants. So what she found is that this, as I said, is a significant inverse relationship between the number of rare non-synonymous variants per individuals and von Willebrand antigen levels. I mean, some people might say, well, that's intuitively correct, but that hasn't been shown before. And after controlling for all the as I said before, age, sex, blood, and ethnicity, still the VWF gene can explain approximately 17% of the variance in the levels. And then if you include this bleeding phenotype, which I'm not going to talk today because phenotyping bleeding, and my colleagues in hematology know this well, is very difficult. If you include the ISTH BAD score, which is this phenotyping tool, you can explain approximately 24% of these. So what we did is to get all these patients that have these 220 or something patients that we have that have low von Willebrand factor levels but have no mutations in the von Willebrand factor gene, and we did a GWAS study. And what she found is like you have ABO and von Willebrand factor, which of course, you know, this is a Manhattan plot that shows all the chromosomes here, and this is exome-wide significance, and red is genome-wide significance. As you can see, there are a couple of other genes that we're investigating that might be associated with this disease. And that's how you have to do this. You know, you get a signal, you try to functionalize it, you try to replicate it in different cohorts, and eventually we'll get there. So we have discovered cohorts and replications, as you can see there. I'm not going to dwell into that, but we have a lot of collaborators around the world that are helping us with this endeavor. And then in the last nugget that I'm going to tell you how we're trying to come across these ethnicity differences, because I think that this is going to be very relevant. So there have been a couple of publications showing, not by our group, by other groups, showing that there are common variants in factor VIII or von Willebrand factor that may influence the levels, okay? But we teamed up with it. This is a nice thing about being in WashU Genome Center with these two people, Ira Hall, who is now at Yale University, and Nathan Stitzel. And we teamed up with them and trying to understand, because they have a large cohort of individuals from African ancestry. And they have whole genome sequencing data on 50,000 individuals. They are cardiovascular genomics. And these cohorts were selected for detection of population-specific alleles. And what do I mean with that? Well, you know, if you incorporate what we call local ancestry, you can, if you sequence, you do whole genome sequencing and can determine haplotype structures of each chromosome of each individual, you can actually tell in this 22 chromosome depiction, you can tell which one, for example, in this particular cohort, which is a haplotype of European ancestry versus African ancestry. And I can show you in the, here in the, on the right side, that there is a gene that has been associated with cardiovascular disease forever, which is the PCSK9 gene. That has been a risk factor for cardiovascular disease, increased lipids. So if you test that gene in this cohort, as you can see here, up there, enough in the African ancestry haplotype, is associated with cardiovascular disease at a much higher rate than if you put us down here in the European ancestry haplotype, indicating that not only the variant is associated with disease, but also the entire haplotype. And why is that? Well, we have I tell you, when we do genome analysis, we have a reference, you probably heard this, the reference genome. So the reference genome, there are actually two genomes, you know. One is James Watson probably, and the other one is maybe Craig Venter or somebody else. But they're two white dudes, you know, like, you know, I'm Hispanic white too. So it's not like, so the reality is that it's a linear genome. So as you can see there, there is only a single haplotype represented at each locus. The relevant sequences might be absent from a poorly represented in this particular genome. And the structural variants might be gone, some of them, because I remind you that the African genome is much more complex because it has been through 200,000 years of recombination, you know, than the European ancestry genome. So what they did actually, and this is not from our lab, is from WashU, part of WashU and Yale actually, and other institutions. They created, this is a great paper for those who love genetics, a draft of a human pan-genome reference. It's a pretty catchy title. And what they did is that they sequenced deep sequencing in large long read sequencing and they created 47 phase diploid assemblies from diverse individuals. So that alone added 120 million base pairs to the so-called reference genome. And they were able, they go to, these are cardiologists, remember, they go to the LPA gene, which is associated with cardiovascular disease. They were able to find association in this population, in this Kringle VNTR domain that has been published 30 years ago, and now they can find it in the African ancestry population. And if you go to this is what it does. If you see in red is the linear genome that we're used to. But if you add all these haplotypes that are from different ancestries, including Asian, African, and so on, you can see that the genome complex, or this locus complex is much more complex, or this locus is much more complex than it used to be with the genome, giving us the ability now to compare apples to apples when we do genome analysis. So what we're doing now, with the genome, WashU Genome Core, I remind you that WashU is responsible for 40% of the Human Genome Project sequencing. So it's a big sequence facility. We have 450 individuals that have this long read sequence. You are sequencing not just the small fragments that you put together bioinformatically. You sequence these long reads that create haplotypes. And eventually we have 120 African Americans, 100, these are healthy individuals, European Americans, Asian American, Hispanic Americans, and we're gonna create for von Willebrand factor and some other loci, we're gonna create this complex reference genome for this particular population. So in summary, I would say that over the last 18 years, we have generated one of the largest repositories for this disease. We have novel genetics association for low von Willebrand factor. We really want to expand these studies to these multi-ethnic cohorts. I think that this is what we owe to our community. And the use of local ancestry can eliminate this ancestry bias that we have developed. And all these pipelines were developed only for that two individuals that have the human reference genome. So going back to case 2, the patient was diagnosed with von Willebrand disease type 1, no pathogenic variants in BWF. The patient was started on estrogen therapy and tranexamic acid. So people in the audience might say, well, I mean, that's nice, all the stuff that you want to do with your genes, but at the end of the day, we treat them the same. We treat the patient the same, which is true. Whole genome sequencing did not show any deleterious variant in other genes that can explain low von Willebrand factor levels. And as I said before, hopefully these studies will allow us to generate these polygenic risk scores to determine risk of bleeding in patients like this one. You know, I don't know if that is just an aspiration, but I think that we can do it if we, if we do what and NIH supports that grant. I wanted to acknowledge people that, as you can imagine, over the last several decades, we've been collaborating with a lot of people, and I'm gonna show more when I talk to the residents and fellows at noon, but I have to acknowledge all the people that collaborated in our platelet projects, as you can see there. And then really, I wanted to acknowledge Bob Montgomery, who has been a great mentor in the field for me, and started this PPG that, again, hopefully is gonna get funded by NIH if you are in the audience. It went through council, but you never know, that's a problem. And we have several collaborators across the world that do it. And finally, I wanted to acknowledge my lab. I couldn't do it without them. I find myself only showing the slides, which is what I do. And they do all the hard work. As you can see there, we have a very diverse and multi-country international lab. And I open the floor for any questions that you may have. I know that we can claim the Cardinals at least. The Rams left, and so we claim the Cardinals. So thank you so much. Thank you so much for your attention. I appreciate it.